In the name of God, and I would like to welcome you to the histology class. I hope you enjoy this kind of teaching also. I remember I taught up to this slide in the last session that we had together. In this slide you see the cytoskeletons. You know, the cytoskeletons have a different component, different parts, and some of them you see here, thin filament or actin, and thick filament like microtubule, and also the myosins. We don't have myosin here, and also the intermediate filament. We call them intermediate filaments because you see the size of the diameters is between the thin and the thick filaments here. We should review this cytoskeleton to better understand the cell adhesions. So let me to go to the cell adhesions. In this slide you see the cell adhesions. If you remember, we said the epithelial cells, each surface of epithelial cells have some features, and we taught the apical surface of the cells features, like the cilium, like the sterocilium, and also the microvillus. Now we are going to teach you the features of the lateral domains of the epithelial cells. In the lateral surface, and the specializations of lateral surface are intercellular junctions. Also we can call them cell-to-cell junctions. You can see some of these junctions here between the two epithelial cells. Also we have these kind of junctions also in the other cells, I mean in the non-epithelial cells, but they are particularly numerous and prominent in epithelial cells. The first one is the tie junction or clothing junctions. These junctions seals between the cells. The cell membranes of the cells attach firmly together, and also these junctions seal the paracellular space. It means the material cannot pass through this pathway. The other name for the tie junctions is zonula occludens. Zonula here means like a belt. These junctions completely circling the epithelial cells and attach the cells to the other cells. Because of that, we call it the zonula occludens. Occludens also means the seals between the cells. The second junction is adhering or anchoring junction. These junctions also strongly attached to cells, to epithelial cell together, but doesn't seal to epithelial cell, the space between the two epithelial cells. Also, this junction is a kind of zonula and like a belt. Another name for this junction is zonula adherens. The third one is the desmosome. These junctions, unlike the two previous junctions, it's not a belt-like, it's not a zonula, it's like a plaque. These cells attach the two epithelial cells in a plaque shape structures. So because of that, we call it the macula adferens. Macula means also like a plaque. The fourth one is the gap junction. This junction is a weak junctions between the arrow junctions. In fact, this junction mediate the intercellular communication rather than the intercellular junctions. It means it forms a pore between the two cells, so material can pass through the two cells or interchange and exchange between the two cells. We call it the gap junctions. The last one is the hemidesmosome. Hemi means the half desmosome, because it's like a half of desmosome. You see, we doesn't connect these junction to the intercellular junctions, and in another box attached to the another box, we call it the cell-to matrix, because this junction is not cell-to-cell junction. It is the cell-to-matrix junctions. And also we have this junction in the basal domains of the epithelial cells, not in the lateral domains, but it's a kind of junction, so we bring it here. Let's see this junction by the shapes, by the image, and explain it in more detail. We have three epithelial cells here. This is the one of them, and this is another cells, and the third one also you see this one, and we have two paracellular space. You can see the tight junction in the apical part. Usually this junction is in the apical part of the cells. Then we have an adherent junction, and then we have a desmosome and also gap junction. You see these tight junctions and adherent junctions are a zanula, are like a belt completely circling the epithelial cells. But the desmosome and also gap junction, they are like a spot and like a block attach the two cells together. Usually we have a tight junction in the apical part of cells. We said the tight junctions seals between the two epithelial cells or paracellular pathway. You can see here two, for example, molecules wants to pass through the paracellular space. And you see here the tight junctions prevent the passing of these molecules from the epithelial cells. By this way, these junctions make a cell between two epithelial cells. So the material cannot pass through this pathway, and they have to go through the intracellular pathway. And as you know, the cells doesn't allow any material easily can pass through the intracellular cells. So by this way, the epithelial cell control entering and leaving of the material from our body. Also, you see the adherent junction is like a belt, I said, but these junctions just attach the two cells together without making any sealing between two cells. This desmosome is like a plaque you see here, also gap junction, and also you see here the hemidesmosome. In the basal part, you see is like half of desmosome. Also, the another molecule stop here and they cannot pass through this pathway. In magnification, you see two cell membranes. One of them is from the one epithelial cells and another is for other epithelial cells attached together by tight junctions. In the tight junction, in fact, the proteins swing and the two cell membranes together, so the material cannot pass between two cells. And these junctions make a lot of strands, branch strands between the two cells. When we have a lot of these strands, the material cannot pass through this pathway. And when we have a few of these strands, some of material when we have a few of these strands, it means these junctions allow or is somehow permeable to materials. It means some material can pass through this way. For example, in the nephrons of kidney, we have a leaking tight junctions. But in the, for example, urinary bladder, we have a unleaking tight junctions. Means we have a lot of these kind of strands between the two epithelial cells. Let me magnify this part in the another slide and see the proteins that participate to form of this junction or zanula occludens. You see we have a lot of proteins. Two important proteins participate to form of these junctions are claudin and occludin. You see one claudin protein from these cells attached firmly to another claudin protein from the other cells, and also occludin like this. And these proteins firmly attach or bring to close two cell membranes, and also for forming a very strong junction between the cells from the inside of the cells, from the cytoplasmic side of the cells. These proteins attach to the cytoskeleton by helping of other proteins like ZO1, ZO2 and ZO3, means zanula occludens 1, zanula occludens 2 and also zanula occludens 3. Attached to the cytoskeleton by this protein, but which kind of cytoskeleton component? You see the actins. In the tight junctions, the protein from the cell membrane attached to the actins, and this attachment causes we have a very strong junction between the two cells. You see the tight junction between the epithelial cells. We have 6 epithelial cells here, and the tight junctions showed here by a red line. You see in the apical part of the cells, and they are also like a belt, or they are zanula encircling around the cells. And the cells procellular pathway, so material have to pass through the intercellular or transcellular pathway. We can see these informations here in this slide. Another function for the tight junction showed in the was showed here, and that is you see the tight junctions between the three epithelial cells. As you know, we have a lot of protein in the cell membrane, and these proteins also can have movement inside of the cell membranes. We call it the cell membrane fluidity. Means the cell membrane component like lipid proteins can move inside of the or within the cell membranes, but tight junctions restrict this movement. You can see the red protein here cannot move to the lateral and basal domains, and the tight junction restricts this movement. And also the lateral and basal protein also cannot move to the apical part. Let me give an example here. For example, these epithelial cells are belong to in your intestine. As you know, the apical protein in the intestine epithelial have a role in digest of materials, digest of foods. So these proteins, for example, are enzyme to digest the proteins. But the other protein here in blue, for example, are protein to attaching the cell to the matrix, to the cell to cells, and have they are a ion channel. So we need this protein in the apical part and this protein in the basal and lateral part. So the tight junction restrict the movement of this protein, and this function of the tight junction is very important and vital in cell behavior and cell life. The second junction was zanula afferens. In the forming of the zanula afferent or zanula junctions, the cadherin protein participate in forming of this junction. You see one cadherin protein from these cells attached to the identical protein from the other cells. I means another cadherin protein from the other cells. You see the E cadherin is the epithelial cadherin, because we have a different kind of cadherin, like N cadherin for the neural cells. Here they are epithelial cadherin protein. The cadherin protein attached together by calcium ions. Without calcium ions, these proteins detach from each other. Also, from the inside of the cell, you see like zonula occludens or tight junction, other protein participate to attach the cadherin to the cytoskeleton. Again, we have a cytoskeleton here, actin filaments. It means in the zanula afferens and zanula occludens, the cell membrane protein attached to the actin filament, or the actin filament as a cell cytoskeleton components. Another picture from the zanula from the afferent junctions or anchoring junctions. You see one cadherin protein from these cells attached to another cadherin protein. In this picture, you see it doesn't show the calcium ions here, and also from the inside this protein by the other protein like catenins, different kind of catenins attached to the cytoskeleton actins. The third junction is desmosome or macular afferents. You can see here the cell membrane from two cells and intercellular or paracellular space. The protein participated in the desmosome junction is also again like atrium junction is caterin. But you can see here we have a special name for this caterin, the desmocoline and the desmoglein. And other protein from the inside of the cells, I mean in the cytoplasm, aggregate and make disc-shaped structures that these structure or these disc-shaped structures attach the caterin to the cytoskeletons here, intermediate filaments. You see the desmosome junction is not like a belt, it's not zanula, it's like a spot like structure, and also this junction also doesn't seals between the two cells, and we have a distance between the two cell membrane. Again, we can see in this schematic picture the caterin proteins. You can see here caterin protein, the desmocoline, and aggregation of some proteins like plakoglobin, plakophilin, and desmoplakin. These proteins make a disc-shaped structures and attach from other side to the intermediate filaments. Again, you see the real picture of the desmosome junction, and also a schematic picture from these real structures by TM. You can see the intermediate filament attached to these junctions. You see two epithelial cells here. One of them is this cell and another is also this cell. They are attached by three junctions, two tie junction and one desmosome junction. These two cells attached together by these three junctions. You can see they are not in order we see in the previous picture. In the previous picture we have first tie junction, then atrium, then desmosome. Here we have a tie junction in the up and down part, and a desmosome in the intermediate part. I think these cells are endothelial cells. We have these cells in the blood vessel. Sometimes the junction between the cells destroyed by autoimmune systems. For example, here we have a epithelial cell of skin, and the desmosome junction between these epithelial cells destroyed by immune cells. You know, this kind of disease called the autoimmune disease. And especially here, immune systems attack the desmoglin or caterin protein in the desmosome junctions and destroy them. So the epithelial cell detached and make a space between the epithelial cells, and this space filled by a liquid or tissue fluid and make a blister. You can see the blister here. And the name of this disease is the pemphigus vulgaris. And the fourth junction is gap junction. As you know, the gap means the distance or the space between two cells. Again, you see here two membranes and paracellular space, and you can see some transmembrane protein attached together. This junction, among the outer junction, is the weakest junction because this junction mediates the intercellular communication rather than adhesion. So because of that, it's a very weak junction. In the formation of these junctions, 6 protein you can see here 6 transmembrane protein called connexin assembled and make a dumbbell-shaped structures, and each dumbbell-shaped structure called connexon. You can see the name connexon. Each protein connexin. One connexon from this cell aligned with another connexon from the other cells and make a pore between two cells and communicate the cytoplasm of these cells with the adjacent cells, so some small molecules can pass through this channel or through this pore. Again, you can see in this picture 6 connexin assemble and make a dumbbell-shaped structure connexon, and 2 connexon from 2 cells aligned or pair up and make a channel between 2 cells so the small molecule can pass through this channel between the 2 cells. We have this junction also in the other tissues, especially between the cardiac muscle cell or the visceral or smooth muscle cells, and this junction in the cardiac cell especially communicate between the cells, and so material can pass between the cells. For example, the calcium can pass from one cell to the another cells, and in a coordinate manner make contractions and make rhythmic contractions between the cells, because the calcium passes through the cells and causes contractions of all cells. You can see here one connexin is very large, it's very resembled to the thread spool. I'm sure you see this thread spool. They are very similar to these connexin. Also have a pore in the middle of these structures. By SEM also we can see the gap junctions. By a technique we call it the cryofractures. 2 membranes detached from each other. We can see this spot. Each of these spots is one connexon. Some cells have a few of them and some cells have a lot of them. It means between 2 cells we have a lot of communications, but between 2 cells we have a limited communication between cells. And the last junction is the hemidesmosome. As you know, and as I said also, it's not a cell-to-cell junction, it's a cell-to-matrix junction. You can see the cell membrane here and extracellular matrix. The cell attached to the extracellular matrix. We call it a hemidesmosome. It's because we have a half of the desmosome here, one plaque attached to the cytoskeleton, again here an intermediate filament, keratin. But as you know, in the desmosome we have a caterin protein, and the caterin protein attach a identical protein from the other cells. But here we have a extracellular matrix, not a cell, so the cell have to change the protein, and the cell use the integrin protein instead of the caterin here, because the integrin can attach to the unidentical protein from the extracellular matrix. We have this junction in the left basal part of epithelial cell. You can see 3 hemidesmosome here. There's also 2 cells here attached to the extracellular matrix by these hemidesmosomes. Like a hemidesmosome, we have another junctions, call it focal adhesion. It's a temporary junctions. Cell use this junction just for a migration, just for a movement. When epithelial cell want to have a movement, especially in a epithelial repair, cell create this junction after the cell finished movement or migration, and this kind of junction appears. It's very similar to the desmosome, but the number of these junction is more than the desmosome. Also, the protein integrin, instead of the attaching to the intermediate filament in the focal adhesion, attach to the actin filaments, and in this actin filament cooperate with the myosin filament and cause the cell contraction and cell movement. Also, peripheral protein from the cytoplasmic side, also in the focal adhesion, is very different than the desmosome, hemidesmosome junction. For example, here we have a paxillin, but in the hemidesmosome we have a desmoplakin, plakoglobin, and other proteins. Let's have a review in this picture. You can see two epithelial cells and different junction between the cells. First, tight junction. This junction, you see cells between the two cells and attached to the actin filament. Adherent junctions, dozen cells, but it's also like a tight junction. It's a zanula, it's like a belt, again attached to the actin filament. Desmosome, it's not a belt, and also it dozen cells between the two cells, and attached to the intermediate filament like a hemidesmosome, and focal adhesion like a tie and adherent junction attached to the actin filament.